All right. So obviously you've heard a lot of the CAR Ts, but everyone's kind of excited. So I'm a little simple. So I get the simplest topic because it's just one thing, right? I don't have to go through all these things that Dr. Chari showed you in the tables and all that. So excellent. So we're in Southern California, right? So you thought you knew cars. I'm going to be basic because I know everybody's at different levels. So we've been talking about CAR Ts, and some of you are really know all of these details, but some of you maybe are still wondering, well, what is a CAR T? So this isn't the CAR we're talking about. This is the CAR we're talking about. So CAR stands for chimeric antigen receptors. And so basically what this means is that these T cells now are getting a new CAR. And this new CAR is a new receptor on their surface that allows them to go and not have to wait for the usual ways the immune system kills cancer. Now they have their own antenna that directs them directly to the cancer cell, okay? So that's what a chimeric antigen receptor means. So how does this work? So usually we use a virus to introduce DNA into the T cell, and then the T cells now can express their CAR on their surface. This CAR then allows the T cell to recognize, for example, BCMA on the myeloma cell, which is the target. And then once it does, once it engages, these CAR T cells multiply, they release cytokines, and they kill the myeloma cell. Okay, so that's sort of the idea behind a CAR T. So there are two approved CAR Ts in myeloma. One is Abecma, used to be called Adicaptogen Viclucel, and the other one is Carvykti. Both are approved for the exact same indication in the United States. So relapse refractory multiple myeloma after at least 4 prior lines of therapy. And you have had to have had the treatments that Dr. Chari showed you, the proteasome inhibitors, so like Velcade, for example, an IMiD like Revlimid, and an anti-CD38 antibody like Darzalex. So KarMMa is the study that got Abecma approved. And so Abecma is a second generation CAR T. So you see the cartoon there. And basically it's binding, the CAR is binding to BCMA. Okay, and BCMA is a very important protein in myeloma, and is basically expressed in all myeloma cells. And so this is the study that was done, and basically patients were treated with 3 doses, 150, 300, and 450 million CAR T cells. And these were patients that were very refractory. They had seen basically everything and had at least 6 prior lines of therapy. So again, patients that had gone through almost all of the therapies that Dr. Chari and Dr. Patel showed you. So this is the response. So if you look all the way to the right, that's everybody's. So when patients are at this, we talked about this a little bit, the expectation is that only about 25 to 30% of patients will actually have a response to a new therapy. So we're seeing much higher responses here. So we're talking about 73% of patients, and we're seeing complete remissions. So that was unheard of before these kind of therapies. So when we're talking about the 20, 30%, they were usually partial remissions. It was like less than 1% or 1% of complete remissions. And now with CAR Ts, we're actually seeing a significant percentage of patients getting complete remissions. And so that's why there's all this excitement. But when you actually look on the left, you see that on the first one's 150, 300, and 450. And the people that got 450 actually seem to be doing better. So there's higher responses with the higher dose of the CAR T. And if you look at the patients just at 450, those patients also stayed in remission longer for about a year or so. So when you get Abecma commercially, that is the dose that we're trying to give you. It's around 450 million CAR T cells. What about Carvykti? So Carvykti was approved based on the CARTITUDE-1 study. And one of the salient features of this CAR T that is different is that you see, I think I have a pointer. Nope, oops, I don't know what I did. Aha. So Carvykti actually has 2 binding sites for BCMA, not just 1. And so we think that because of that, perhaps it actually more avidly binds to the myeloma cells. And so again, this was about 100 patients were enrolled, and here they only gave 1 dose. So they didn't have that dose escalation that we saw with the other trial, but patients also heavily pretreated, median 6 prior lines of therapy. And these are the responses, so quite impressive, right? So basically almost all patients responded, And a lot of patients were in complete remission or better, 80%. And so the latest time we looked, it's been 27 months since the study started, and we have not reached the median, meaning that more than half the patients are still in remission and obviously alive. So it's looking quite good. So that's what got this CAR-T approved. So with new therapies, we get new toxicities, right? So these two I put up here are sort of the main toxicities that we worry about with CAR-Ts. And also you may have heard about, I was doing questions, so I didn't get to see Dr. Patel's talk, but she may have talked about these toxicities with the bispecifics, but if she didn't, we'll talk now. So we're talking about cytokine release syndrome and neurotoxicity. So anytime you engage the immune system, you're going to get side effects. So when you get the flu, right, you get fevers, you get achy, and you feel kind of crappy. That's actually not the virus. That's your immune system fighting the virus that is causing those symptoms. If you get a really bad infection, you can get this condition called sepsis, right, where it's very dangerous, where the immune system is so wrapped up that you start sort of getting fluid in your lungs and your blood pressure drops, you end up in an intensive care unit. Again, that is not necessarily the infection, it's the immune system that is so active trying to get rid of this bad infection that is kind of leaving collateral damage. And that's what cytokine release syndrome is. So when you give the CAR-Ts are expanding. And as they're expanding, they're sending all these cytokines that are leading to these side effects. Luckily, for most of these therapies, we're talking about more like flu-like symptoms than true sepsis. And luckily, we also have medicines that can reverse the process. We just need to recognize it and intervene right away, okay? So that's cytokine release. Neurotoxicity is a little bit similar, although we don't understand it as well. But when all these cytokines get released, you also end up sort of affecting the blood-brain barrier. And so you start getting sort of the cells that go in there and causing inflammation. And so neurotoxicity, or ICANS, can be as simple as perhaps some mild confusion, maybe some mild word finding, like you can't quite find the word to say, kind of like I'm doing now, right? So you can't find that word and you notice sort of some off things, but then it goes away. But it could also be severe, where you can actually get stroke-like symptoms, seizures, and even go into a full coma. So it can be very dangerous. And so those are called grades of toxicity. Luckily, with CAR-Ts, for the most part, they tend to be, again, the low grade. And as long as we intervene early, we can stop the process and it doesn't become any worse. But obviously, these are, if left untreated or if they progress beyond the treatments, they are life-threatening. So when we talk about the cytokine release and the neurotoxicity, for the most part, we're thinking early on, so the first couple of weeks after the CAR-T. But one of the things that I think we don't talk enough about, that we actually mentioned several times in the panels, was this cytopenia. So when people get CAR-Ts, your blood counts drop like when you get chemotherapy. And it's especially true when your bone marrow has a lot of myeloma cells. So the CAR-Ts are so powerful, they go in there and they kill the myeloma cells in the bone marrow, that they actually cause a lot of damage, and the bone marrow takes a long time to recover. And so that's often when we see these sort of prolonged cytopenias. Also, before the CAR-T, you get chemotherapy that also can cause sort of your blood counts to come low. So it's a combination of two things that happen. And so it's very common to get like low neutrophils and low platelets, and often require transfusions. But in about a third of patients, this continues into later, beyond a month. Sometimes even 3 months later, people still have low blood counts. And so that's when we're talking about maybe giving stem cells to try and restore that. But luckily, that is rare. Most people eventually do recover their blood counts. But because of this, one of the important things that we need to watch out for is infections. So not only are your blood counts low, we gave you chemotherapy to tamp down your T cells. We gave you CAR-Ts that engage your T cells, and that's the part of your immune system that protects you from viruses and things like that, right? So those will be low. The CAR-Ts are going after your plasma cells, and those are the cells that make your antibodies. So you got rid of those too. So now you have basically no B cells, and you have low T cells. So infections and infection prevention is incredibly important after CAR-Ts. All right, so what does it mean to go through CAR-Ts? So the CAR-Ts that we're talking about that are approved are what we call autologous CAR-T, meaning that they're made out of T cells from yourself, okay? If you're a patient, of course. And so what does the patient go through? So the patient first has to be collected. So those T cells are collected and through apheresis. So similar to stem cell collection. And we'll talk about the difference from an autologous transplant in a second. So you collect your T cells, and then those T cells have to be manufactured, right? You have to make the CAR Ts. Well, you send the T cells and that manufacturing process, and by the time you get them back, it could be about 4 weeks. Here it says 10 to 28 days, but we all know that it can be 1 to 2 months before we get them back. So during that time, before you actually get your CAR Ts, we need to make sure that your myeloma is under control and that it doesn't continue to progress. And so a lot of people will need what we call bridging therapy to keep the myeloma under control before we can give you to get the CAR Ts. And so once you're ready to get CAR Ts, then you get this low dose of chemotherapy, which is called lymphodepletion, that allows your CAR Ts to expand, and then you get the infusion of your CAR Ts. So the process itself actually is very similar to an autologous stem cell transplant in sort of the logistics, but with very special differences, right? So when you do an autologous stem cell transplant, you first have to collect your stem cells. And so we talked about that process where you have to stimulate those stem cells to leave the bone marrow to start circulating. And then you go through the leukapheresis where your blood goes to the machine and we collect those stem cells and put them away and we freeze them. Once you collect though, it's only about a week or 2 weeks or less, you come in and actually get your transplant. And the transplant really is just giving you high doses of melphalan. It's the chemotherapy that is the treatment. And then you get the stem cells just to rescue you from the chemotherapy. And so the toxicities that you then experience are from the chemotherapy, not from the cells, right? So then once your blood counts recover, you kind of leave the hospital. If you're in the hospital, you get kind of leave the local area. With CAR Ts, you go through that collection again, but you don't have to stimulate the T cells. The T cells are already circulating in your bloodstream, so you don't have to go through all that mobilization. So it's a much faster process, an easier process. So you collect T cells, but then you have to send them away to manufacture the CAR Ts. And we talked about how that takes a long time. During that time, you might need bridging therapy to help control your CAR Ts, because again, a very important distinction here is when we do a stem cell transplant, you're in remission, right? You've gotten your induction chemo and you're under control and you get your transplant. With the CAR Ts right now, the way we're giving them, you're progressing, the disease is progressing. So we need to kind of keep that from becoming an issue before we give you your CAR Ts, right? Because if your myeloma progresses, it could hurt your kidneys and make you ineligible for the CAR Ts. So you need to get bridging therapy. And then once you're actually ready to get the CAR Ts, you get this low dose of chemotherapy. And the purpose of the chemotherapy is not to treat your myeloma, is to basically sort of tone down your normal T cells. So when the CAR Ts come in, they can expand and go after your myeloma. But it's the CAR Ts that are the treatment here, not the chemotherapy. And so the toxicity is not from the chemotherapy necessarily, although it adds a little bit, it's from the CAR Ts. So similar in process, but very different in terms of the actual treatment. All right, I was asked to predict the future. So before I can predict the future, I'm going to talk about sort of what are the shortcomings right now with CAR Ts, right? So the first part is improving the efficacy. So you're probably saying, well, 100% response is pretty efficacious, right? So how can we improve on that? But the truth is that the majority of patients are still relapsing after CAR Ts. So unfortunately, CAR Ts don't seem to be curative the way we're giving them right now. So obviously that needs to change. So we need to understand why. Oh, here I go again. There we go. We need to understand why that's happening. And so there's a lot of studies going on and trying to figure out the mechanisms of resistance to CAR Ts. And then the question is, will they work better earlier, right? Dr. Chari sort of brought it up in his talk about should we be giving CAR Ts even in the frontline setting? And so those studies are ongoing at this point. And then, right now, the nice thing about CAR T is that it's a one and done, right? So those of you who may have had a CAR T, that's probably what you enjoy the most, that you're not on a maintenance therapy of any sort and you're kind of getting a break. But do we need to give maintenance perhaps in some patients to maintain that remission? And again, some of those studies are ongoing. Hopefully not, or at least not for a long time, we'll see. Can we do anything to improve the quality of those T cells, right? So we know that the the effectiveness of the CAR T depends on the substrate. So if the T cells we collect don't work very well, the CAR Ts are not going to work very well either. So is there something we can do to actually help them out? And can we go after different targets? We need to make them safer. So we need to sort of identify why some patients get high-grade cytokine release and neurotoxicity and why others don't. We know that, for example, when you come in with bulky disease, you're more likely to have higher grade of symptoms. And so now we're getting better at maybe controlling or kind of tamping down your disease so that those toxicities are not as great. There are some CAR Ts that have safety switches so that if things get out of hand, you can actually go in and actually you basically commit suicide and they go away. So things like that hopefully will make this an easier, more safer treatment, because that's what's going to be required if we're going to be looking at giving it at the very beginning, right? When people can live for so many years with myeloma, you don't want to give something that could potentially harm you. So, and then access. So I don't know who's actually gotten commercial CAR T, but it can be a difficult process, right? There's not enough. I don't think the current CAR Ts are enough for the demand that is out there. And so there's people that have been on wait lists for 6+ months and to get these CAR Ts. So that's a problem. It also takes a long time to get them back. And so sort of kind of making CAR Ts faster, that's going to be an important process. And so perhaps these allogeneics or off-the-shelf CAR Ts may help with that. And then these are really expensive. So Carvykti is, I think, $457,000 pop just for the actual CAR T. And so these are pretty expensive therapies. These are all just a list of all the studies that are ongoing in earlier lines. So looking at CAR Ts sort of beyond that relapse refractory setting and even in the frontline setting. So stay tuned for those. And then I think we have sort of new and improved CAR Ts that are coming. So we have alternate targets. So the 2 CAR Ts that are FDA approved are against BCMA. And then you heard with the bispecifics, we have other targets like GPRC5D and CS1. And so those are ongoing. And you'll see actually some of you here may have already gotten GPRC5D because I know City of Hope has a study with that. And so, should we be doing multi-targeted CAR Ts? Can we go after different sort of proteins? Kind of like we do that already, right? We do combination treatment in myeloma all the time. So can we do it with just, one T cell going after different things? For example, we talked about off the shelf. So your T cells, if you're a patient, have had a lot of chemotherapy, maybe they're not working as well anymore. So maybe we should get a donor that doesn't have cancer, hasn't had chemo, and use their T cells to do CAR Ts. And those are those allogeneic CAR T cells. And then this concept of an armored CAR. So it's kind of like, I think of it as a CAR T that has its own pit stop crew. So basically these are CAR Ts that can secrete cytokines that can help them revitalize and persist. And so basically they're providing their own fuel and changing their tires, et cetera, to hopefully keep them moving longer. So all those things are going on. And so just really quickly, I don't know how much time I have. I'm good. So I'm not going to go through this trial, but this is the first allogeneic CAR T. So this is using T cells from a donor that doesn't have cancer or have had chemo. And so it's called Allo715. One of the things is to do that, you have to be more careful because, when you get a transplant using someone else's T cells, you have to match, right? So that those T cells don't cause graft versus host disease. So here, the technology has improved enough that you can actually remove parts of the T cells that usually sort of mediate graft versus host disease so that T cells can't do that to the body. And so far, the allogeneic CAR Ts have not caused graft versus host disease. So stay tuned. Early data, they look active. Question is, can they persist as long as the autologous CAR Ts? We now have a new target, GPRC5D, so CAR T that looks really good against this new target. And so we now have patients who've gone from a CAR T against BCMA to a CAR T against GPRC5D with good results. So again, stay tuned for more of that. There'll be a presentation at ASH this year of a GPRC5D CAR T as well. And then we have these CAR Ts called FastCAR, which is a dual targeted CAR T. So it targets, we talked about sort of going after 2 different sort of proteins. So in this case, BCMA and CD19. And the plus of this is also that they can do very quick manufacturing. So they say next day manufacturing, although because of all the regulatory tests and so forth, it still takes about 10 to 14 days to actually get the cells back. But again, it looks like a very promising CAR T. So CAR Ts are powerful, right? They work against myeloma. It's a one-time treatment, at least right now. So that's a really nice thing. Clearly very logistical specific issues that we haven't seen with other type of products. So it does limit the access, unfortunately. It may work better if done earlier, and that's being tested. And so it is a very exciting therapy. So I guess the question is, what do I see for the future itself? I see the CAR Ts. I think CAR Ts will be used much earlier. And I even think it'll be in frontline. We're starting with the high-risk patients, but ultimately I think all patients will be getting potentially CAR Ts as their frontline, unlike what Dr. Chari said. I think we should be using CAR Ts as consolidation. I don't think we should be giving CAR Ts when the disease is rampant and growing. I think these are very powerful. We don't need a lot of disease to help them expand. So I see it as somebody getting, for example, induction, getting remission, give them a CAR T, just like we do with transplant. And what we've seen, and you'll see some data, is that when we do that, there's much less cytokine release. They seem to be much safer, and they work better, actually. And so I ultimately see that we will be using CAR Ts, hopefully with some of our other exciting therapies, where I anticipate that people will get some kind of induction to control the disease, get the CAR T like we do transplants consolidation, and then follow with like a 6-month of a bispecific, and then actually stop. And that's what I see for the future, and that's what I see especially.